Well, thanks, Jerry. That's a very kind introduction, and it's a real pleasure to be here at the Simons Foundation. And I want to thank the Simons Foundation for their support of Autism BrainNet, because you can have a million good ideas, but unless you have the support to make them reality, they're just good ideas. And the Simons Foundation and Jerry has really been leading the effort to convince. I convinced him, but then he convinced everybody else that this was a good idea. So we really appreciate his strong support. So I'm going to dig right in because I've changed this lecture quite a bit. I'm not sure exactly how long it is, so I want to talk about our efforts to try and understand the brain changes or alterations that are typical of autism. And right from the get-go, you can imagine that it's going to be complicated because autism itself is complicated. So not only are there different domains of behavior that are affected as core features, so social behavior and repetitive behaviors, which would imply that there are different brain systems underlying both of these. But of course, there are these numerous comorbid conditions that are increasingly becoming more and more a definition or part of the definition of autism. So many individuals with autism have developmental delays. Anxiety may occur in as many as 85% of people on the autism spectrum. There's digestive disorders in 30% of people with autism, and again, 30% of people with autism have epilepsy. All implying that there are different brain regions that might be actually involved in mediating these different problems that people with autism have. In terms of what causes autism, well, we still don't know, but we know that it's, again, very complicated. Many genes, there's now a couple of hundred genes that have been identified that are risk factors. But none of them are so deterministic that they absolutely cause autism. In fact, sometimes you see that they're common across autism and schizophrenia and other psychiatric disorders. So now the notion is that you can have genetic risk factors, but you also have to have an environmental impact in order to have autism. And I think that there's increasing evidence that even on a background of normal genetics or genetics that are not at risk for autism, you can have environmental factors like viral exposure during pregnancy or other kinds of things that could lead to autism. So we have been thinking that there's many types of autism. I used to say when I give a general talk that there are likely to be many causes of autism, but I think we're absolutely clear that there are many causes of autism. And so the implication is that there may be different trajectories of developmental alterations in autism that you may see. We're not expecting to see just one pattern of brain changes that is typical of all autism. So one of the fun things about coming to the Simons Foundation to give this presentation is that I went back and reviewed many papers that have used MRI, magnetic resonance imaging, to study living people with autism. And these studies started about 1988, 1987. And if you look at the whole history of MRI studies, there's a number of weaknesses that decrease what we can take from them. So many of them, for example, have very small sample sizes. Some of the earliest studies had just 15 or 17 or 18 people in them. The samples are often very heterogeneous. So you'd have people as young as 3, but as old as 30 in the same study. And we know this is a developmental disorder, so the brain changes over time. We imagine that the disorder is going to be changing over time. Many of the studies were cross-sectional, so they compared different groups of people at 10 years of age with people at 20 years of age and try and draw longitudinal assumptions, and that is often dangerous. And by and large, the vast majority of imaging studies have been done on older individuals with autism and higher functioning individuals with autism. And the reason for that is it's hard. You have to stick a person in a tube. They have to stay still for 30 minutes. And if you're not compliant or you're not sedated, it's very hard to get those images. So that's why the vast majority of of studies really are older and higher functioning individuals. So I'm going to try to make the case that it's important to study very young individuals with autism during the first several years of life. And I'm going to take a diversion to give you an example of how in my own thinking I've come to this conclusion. So as Jerry said, we have studied basic neurobiological questions in the past, and one of them is we've studied parts of the brain that are involved in memory. So we've done studies where we work with rhesus monkeys, and we've tried to figure out what parts of the brain are involved in episodic or declarative memory, memories that you would take home from this session today. And one of the studies that we did was to carry out what's called a spatial memory task. The animals would be let into a room that's a hexagonal room, and they'd search for food. And the food would be under cups that are located where you see those colored bars or colored circles, okay? So you'd have the environment around this hexagonal maze complicated so that the monkey, even if you didn't put the colored cups there, the monkey eventually learns where the goodies are. And they do that by navigating in space. They know that the colored cups or the uncolored cups where the goodies are in a certain position relative to things like the flag and whatever. And this is the typical kind of spatial memory task that the hippocampus is essential for. And so just to show you how this works, this is first of all a monkey who has his hippocampus. He's been in this task for a long time, and he's going around, and this is the maze, and now there's no colored cups. They're all colorless, but he's looking for the goodies. And he got some initially, went over to another place, got some. Now he's looking around a little bit and trying to figure out where the remaining goodies are. And you'll see him, so he's just thinking about it, looking around the room. And eventually, he walks over to where he thinks the other goodies are, skips some cups, and finds the goodies, okay? So he knows where they are in this maze, and he's got a very good spatial memory of this maze. At this point, he stops working because he knows he's gotten all the goodies. Now I'm going to show you a monkey that doesn't have a hippocampus, okay? 1 second. So he's ready to go because he knows he's not going to get any more cookies. And here comes a monkey that doesn't have a hippocampus. Surgically removed, right. Both sides. This is a bilateral hippocampal lesion, right. This monkey doesn't have a clue where the goodies are. What he's doing is randomly going after the cups. And this monkey will never be able to learn this task. We know from human subjects who've had similar bilateral removals of the hippocampus that they cannot make a map of external space, and they never will be able to do that. So what was interesting when we did this study is we had one monkey who was a control monkey who couldn't do the task. And we thought, this is odd, what's going on? So we did an MRI of this monkey. And at the level of the MRI, his hippocampus was there. It's actually where the arrows are. You can see his hippocampus. But only when we actually did the microscopic histology of this animal's brain did we realize that where the arrows are, this monkey had had a bilateral ischemic lesion of his hippocampus. Really tiny, but it went throughout the entire extent of his hippocampus. And we looked at the rest of his entire brain, and it was fine. It was this lesion that caused this animal to have an impairment on this memory task, okay? So this just shows how sensitive this spatial memory task is to a hippocampal lesion. This was about 3 or 4% of the hippocampus that was damaged in this case. Okay, so here's the real point. So then we do the same study, but now we do the surgical resection of the lesion in a 2-week-old monkey. This is basically a newborn monkey. And we tested them at several times during the development. But what I'm going to tell you about is when we tested them as adults, okay? Now at the same age that you just saw those monkeys. So here's the lesion. So this we actually purposely tried to remove the hippocampus. You see a control monkey up top and down below. That black hole, that's not photoshopped. That's actually just the ventricle that's enlarged because the hippocampus is gone. And this animal, his hippocampus is completely gone. In all the animals in this study, actually, the hippocampus was completely gone. So we put them in the maze, and one thing that we saw with these animals is that they were hyperactive, which is typical of monkeys that don't have a hippocampus. But imagine our surprise when we tested them on the spatial maze, and they were completely normal. They could do the task exactly as well as control animals, right? Okay, so the difference is that these animals had their hippocampus removed at 2 weeks, and then had a lifetime of experience and brain plasticity that modified their brain and allowed this animal to accomplish this task using parts of the brain that are different from the hippocampus. Now, Jerry said I'm a neuroanatomist. I'm a neuroanatomical and structure chauvinist. I always used to think that the hippocampus was designed by evolution to do these kinds of tasks. If you took it out, the animal wouldn't be able to do it. It turns out that's wrong, and I've now completely changed my view, particularly when you have brain damage in young animals. The brain can reorganize itself and can accomplish behaviors that in an adult with the same lesion wouldn't be able to accomplish. So what does this have to do with ASD? Well, first of all, I think we make the assumption that in autism there's been an insult very early on that leads to some modification of the brain. Not only this work, but many lines of work suggest that the damaged brain tries to compensate and to optimize its ability to deal with a complex environment. So the longer one waits postnatally, the more likely it is that alterations of brain structure and function are due to compensatory changes rather than the original insult. And this has probably made all the worse, or all the better in a sense, because behavioral therapy and drug treatments probably enhance brain plasticity. So the bottom line is that when you look at the brain of a 16-year-old, you're no longer looking at autism, but a brain that has had 16 years of environmental experience to adapt and compensate to having autism. So how this relates to the imaging literature is, again, the vast majority of imaging studies are done on teenagers or adults. My view would be, that's all well and good, but you're no longer looking at the pathology associated with autism. You're looking for the pathology that has gone through this compensatory process, and you're looking at plasticity now, not the root causes of autism. So this was just a PubMed search that I did. Out of the 1,362 papers that you can find on Medline that are related to autism and MRI, probably 20 or so have actually used subjects that are younger than 5 years of age. And so there's still, in the literature of autism, only a small number of studies that have dealt with this very young population. So I'm going to tell you about some of our studies that actually has tapped into that very young population. And given everything I've told you so far, our hypothesis is that you're not going to see one common pathology. You're going to see a variety. And we're going to try and do what we can to sort of figure out what that variety is. We do that in a study called the Autism Phenome Project, started in 2006. It's a multidisciplinary project. We recruit kids when they're 2 to 3 years of age. The study includes all children with autism. We have very few exclusionary criteria. We have included both boys and girls in this study, and we have a smaller number, about 1/3 of the kids are age-matched controls. It's a longitudinal study, so the children return to the Mind Institute annually. And in addition to testing the children, we take blood samples from them and from their siblings and from their parents to carry out genetic, immune, and other kinds of studies. The first year of the study, it's a very intensive study. The families go through these 5 sessions. I won't go in detail with everything that we do, but they're re-diagnosed with our clinicians. They go through extensive medical and behavioral assessment. We collect all their medical records, which allows us then to go back and look retrospectively at some of the things that happened to them early on. What I'm going to focus on is visit number 4 when we do an MRI study with the children. But before I get to that, just to give you some of the facts, so we've had 366 families in this study. You can see the number of boys and girls. We actually recruit at the prevalence rate of autism, about 4 to 1, but we have about 35 typically developing girls and 42 girls with autism in the study. They, on average, are 3 years of age when they come in, and the range is between 2 and 5. We've tried to sort of push it towards the younger ages, but the average age is 3. It's diverse. We're in California. We try and match our population, so it's ethnically and racially diverse. When you recruit kids this young, they tend to be more severely affected. So this is just one measure of autism severity based on the ADOS. Below 4, you wouldn't have a diagnosis of autism. What you can see is the kids in our study tend to be biased towards the more severely affected. In our study, like most other studies of kids this age, we find that the average IQ or developmental quotient is in the 60s. So the kids, even though the range is really broad from verbal developmental quotient of 20 to 150 or so, the average is around 60 or so. So this is a population that is developmentally impaired. So the first thing to tell you is how do we actually scan these kids? So many studies of young kids with autism will sedate the kids. And you can actually get IRB approval, ethical approval to do that. And we did as well. But we actually did focus groups with the families and asked them, how do you feel about having your child sedated? And all of the parents, virtually all of the parents who had children with autism said, yeah, we'll do that one time. But we're not going to come back over and over again to do that. And then, of course, we can't do sedation with the typically developing kids. So we decided to go a different route. We actually worked with the families. And in the end, this project is successful because we've been intensely involved with working with the families. So what we do is 2 weeks before the scan, actually on our website, we have videos about how the scan is done, and it educates the families about what they can expect. And then 2 weeks before the scan, the children and family come in. This is a mock scanner, the thing that top left. So it actually makes all the noises of a scanner, and the child can go into the tube and whatever. And so we allow the child to play on the scanner and to experience it. And then we send the family home with a packet that includes those earphones. How many people have had an MRI? Yeah, wow. At the MRI store on the corner, right? Most people have experienced how noisy it is, and we obviously have to do ear protection. And this was, even this is a challenge, right? Because children with autism have tactile sensitivities. So actually to put earphones on them is not trivial. So 2 weeks beforehand, we send the earphones and earplugs home with the parents and a CD that has the gradient sounds of the MRI. So every night the child will go to sleep, and then the parent will sneak up on it and put earplugs in and put earphones on, and then all of a sudden puts a CD in the boombox and boom, 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 the gradient sounds. And in those that are successful, day after day, the child stays asleep more and more and more. And by the time that they come into the scanner, hopefully, we've habituated them to all of the nastiness of the MRI. So on a good day, well, here's one of our children practicing right here. And we've done this now with kids, I'll tell you at the end of the talk, as young as six months, and it works very, very well. So on the day of the scanner, not only do we train the families, but we also change the environment. So on the left is what our MRI looks like normally, and then we make it child-friendly and family-friendly. We actually put MRI-compatible gurneys next to the gantry, so that it turns it into a queen-size bed. And then the mom or dad or both can get on the gantry and read to the child. Whatever they normally do to get the child to sleep, we can pipe in videos, we can play songs, whatever. And the child will go to sleep. On a good night, it takes a half an hour. On a bad night, it takes 3 hours. But fortunately, we can be patient and take whatever time it takes. The child then will, when they're asleep, will be put into the head cage, and then for every scan that we've done, we always have assistants who are watching the child for the entire time that they're being scanned, because we don't want the child to wake up and be frightened. So if the child even stirs, we can pull the child out immediately. If they go back to sleep, we put them back in and continue. But again, this has been, when we got into this, our colleagues were saying, well, maybe if you're lucky, you'll get 50% of the kids that will be able to endure this. We have a terrific team. I want to acknowledge particularly Christine Nordahl, who's in the front there. This is our scanning team. They all have bags under their eyes, but they're a terrific team, very, very patient. And in the end, we've been enormously successful. So that you can see, here's our success rate: time 1, 88%, time 2, 90%, and time 3, 87%. Again, a year older at each of these time points. And this is really a testimony to the MRI team, who really know the families. And I have to say, I've had the privilege of being able to do some of this scanning. And the time that we spend waiting for the child to go to sleep has been enormously beneficial to me, because I get that time to talk to the parents and learn about autism. I mean, this is how we've learned about autism. So now I want to tell you about some of the findings. And I'm going to start by saying that the field of autism research has been influenced highly by work that was done by Eric Courchain starting in 2001. And they published, this is with a student, Elizabeth Redkay, published a review paper that basically showed that this curve, the yellow line, is showing the size of the brain relative to normal brains, which is the dotted line, of kids with autism from different studies that have looked at populations at different ages. And what it basically says is that the brain of children with autism is too big early on. Okay, so during the first few years of life, the curve goes way up to about 10% larger than normal, and then it sort of comes back down to 0. Why it comes back down to 0, we don't have a clue what's going on there, because nobody's actually ever done a longitudinal study to look at a group of individuals from very early on to very late on. So we don't know what's going on there, but I'm going to focus on this early period. So first of all, let me tell you about whether we replicated this in the Autism Phenome Project. And we did. So if you take all of the kids in the project that we scanned and you look at their total brain, and we've done all the bits and pieces of the brain, and I don't have time to go into all of that, but I'm just going to try and give you the big picture. The total brain in autism, it's in red, is about 6% larger in boys on average than in autistic boys and in boys that are typically developing. One of the interesting things is that we actually don't find a difference in girls. Now, again, it's a smaller sample, but there's increasing evidence that autism in girls is very different from autism in boys. And everything that we've seen so far in terms of our imaging studies would suggest that that's true. So here's the first thing. We don't see, other than I think 2 girls in our entire sample, we don't see many girls with a very large brain. But the other thing that impressed us, and again, coming into these studies, you would think that all brains are too big, that all kids with autism have a brain that's too big. But we had a big enough sample that we could actually say, well, what is the percentage of kids with autism that have a brain that's too big? And it turns out this is just a scatter plot of typically developing kids in our study in blue, and then the kids with autism. And what you can see is that there's a lot of overlap, right? But there's a group of kids with autism, the red dots up there, that have really big brains. There's some that even have really small brains. And we started asking questions about these kids who have the really big brains up top there. First question we asked is, are these the kids who have the early onset form of autism, or are these the kids who have regressive autism? So in our study, and again, this was something that was controversial when I first got started, that there even was regressive autism, but I think everybody sort of accepts that now. And the bottom line is, so regression is that if you look at a child at 12 months of age, the early onset child is going to start showing some difficulties already. You wouldn't call it autism, but they're going to have some developmental delays. And clearly there's a problem. A clinician would say something's going on with this child. Whereas if you looked at the child that has regression at 12 months, they look fine. They're sociable, they're gaining language, and then sometime between 18 and 24 months, something happens and they regress into autism, okay? So they lose abilities. So, an interesting dichotomy. It's actually more complicated than that, and we were curious whether it was the kids with the early onset form, which is what we predicted, that had the big brains, or the kids with the regressive form, which we thought probably didn't have the big brains. So we were wrong. It turns out that it's the kids in green here that have the regressive form of autism that have the big brains, by and large. 88% of kids who are characterized as regressive have the large brains, the megalencephalic brains, okay? There are some kids with the early onset, but the percentage is much, much lower. So this was sort of interesting. The kids with the regression, so you don't actually see the behavioral signs and symptoms of autism until the kids are 18 to 24 months. They have this big brain, and we are curious, when did the big brain phenomenon begin during their lives? When could you actually detect it? And because we have all the medical records, we could actually go back and look at head circumference. And it turns out that the head circumference absolutely replicated what we were seeing in the MRI. So in this chart, the blue line is the growth of the head circumference for the typically developing kids. The dashed line is the head circumference for the early onset kids. And the solid red line is the kids who had the regressive autism. And interestingly, they started departing from normality around 4 to 6 months of age. Okay? So their brains started getting too big at 4 to 6 months of age, even though behaviorally you didn't see any problems with these kids to 18 to 24 months. Okay? Interestingly, as a side effect, the last line in this paper that we've reported this, it occurred to us that many parents are concerned that the regressive form of autism is started by one of the vaccinations that are taking place at 18 to 24 months. But actually, these data argue against that. These would suggest that the biological process that ultimately leads to regression into autism started at least by 4 to 6 months of age. Okay? So the bottom line from this part is that the vast majority of kids with autism, their brains, which one would be in the middle here, the early onset form. If you look at, this is a child, the typical child is 31 months, the child with autism is 32 months. The size of their brain is almost identical. And as a trained neuroanatomist, I couldn't pick that brain out as having autism. But there's a subset of kids, the one on the far right, you can see almost 200 cc's larger than both of these brains. And that one you could actually probably pick out and say something's abnormal about that brain. So there has been this issue that's come up lately about whether the kids with autism have big brains just because they're bigger. And so it is true in the literature, kids with autism tend to be larger. They tend to be taller. It's not been all that well studied, but there is sort of a kernel throughout the literature suggesting that kids with autism are bigger. We have looked at that in our population again. And what you can see up top here, top left, this is actually plotted the height versus the kid's age. And what you can see is that there are some kids with autism, the red dots far up, that are taller than the typically developing kids. And it turns out, though, that on average, in our Autism Phenome Project, because of the overlap of kids, there is no significant difference in terms of height between kids with autism and typically developing kids. And moreover, in this plot, what I've done is the kids who have the big brains are color-coded in green. The kids who have the normal-sized brain, these are all kids with autism, the normal-sized brain are shown in red. And the kids, remember there was a subset that have a brain that's too small. We've color-coded them as gray. It turns out it's the kids who have the small brains who are tallest. for reason, who would know? But until you actually look at the data, and a large enough sample where you can actually have 7 or 10 kids that have the small brains, you wouldn't be able to see this kind of thing. So in our study, at least, it's not that the kids have a big brain just because they're bigger kids. Actually, what you see is the green dots there. Those are the kids with the big brains. They're right in the middle of the size of the typically developing kids. And that's what we see with the statistics as well. There's no difference. Okay? So this issue of macrocephaly, and macrocephaly is big head, megalencephaly is big brain. It's something I don't want to get the message that this is only occurring in autism, because if you look online in the Mendelian Inheritance in Man database, you'll find that there's lots and lots of syndromes that are associated with macrocephaly. But I do think it's a phenotype of autism and worthy of additional study. So we actually now use this formula for talking about kids with big brains. We call it disproportionate megalencephaly, where the ratio of brain volume to height is 1.5 standard deviations above the control. That's just a limit. You have to draw a limit someplace, and we're looking at these kids and trying to determine whether there's behavioral differences and whether there's other differences as well. This subphenotype in our study constitutes about 15% of kids with autism. Okay, so the big brain phenomenon does exist, but it's a minority of kids with autism. It's actually 15% of the boys. Still not clear how many of the girls have the big brain phenomenon. So there was a paper that came out last year, basically that made the claim that all the studies on macrocephaly were wrong and were overstated because they highlighted studies that looked at kids with autism now and compared their head size to the CDC standards. And what they basically said was that was inappropriate, because even if you took typical kids now and compared them to CDC standards, you'd show this early increased head growth. And so I think that that's a valid point and something that needs to be considered. But I think what people took from this paper is that the big brain form of autism doesn't occur. And that's wrong. Our data suggests that it occurs. It's a minority. There's been very nice data from Heather Cody at UNC with Joe Piven's group showing, again, using local control subjects, not the CDC controls, that there is a population, or that there's enhanced brain growth early on in kids with autism. And there's actually studies all over the world that have shown this. This is a Japanese study that compared, 85 kids with autism to 14,000 contemporary Japanese typically developing children, and again found that there was enhanced head growth, brain growth over the first year of life. So again, I wanted to mention this because things swing, and sometimes they swing too far, and I think this is an important subgroup. It does exist, it can be demonstrated, but it's hard to demonstrate it when you have very small sample sizes, because again, it only constitutes 15% of the population that you're going to see out there. So you would think, okay, this is cool. You have 15% of the ASD population that has macrocephaly. They must all have the same genetic mutation, right? And turns out wrong. So this is a nice attempt by people at UCLA to look for the genetic underpinnings of macrocephaly. What they found was that about 22% had a mutation in a gene called PTEN that's been associated with macrocephaly, but only 22%. And then there was a potpourri of genetic mutations that were seen in all the other kids with macrocephaly. We've been collaborating with Evan Eichler and others to look in our group of 19 kids with megalencephaly, and the same thing. We see not a single pattern of genetic mutations that are associated with this. We're still working on that, but it doesn't make it all that much easier to have this subphenotype. I'm not going to go into it. We've been looking at the behavioral characteristics of this group. So far, nothing dramatically different between the big kids that have, or the kids with autism that have the big brains and the other kids, but we're still looking. So one of the mysteries in my mind now is what causes the brains to be too big? And this is just an illustration. This is a stained section. This is actually from a group of beautiful monographs by a guy named Cornell who studied the developing human brain. And each black dot is a neuron, and you can see that in a 1-month-old child, it's really very dense, and that the density decreases and decreases, so that by 24 months, the space in between the black dots is larger. And that's because these are neurons, and the neurons are becoming more mature over that period of time. But the question is, what is it in the maturation of the brain that leads the brain to be too big? Is it that there's too many neurons? Are the neurons too big? There's another group of cells in the brain called glial cells. Are there too many glial cells? Some people have suggested that it may be that there's an inflammatory process going on and that there's too many glial cells. At the moment, we don't know, and I'm not sure that imaging is going to be able to allow us to answer that question. Here again is a critical need for having postmortem material. It really, we're only going to be able to answer that question when we can look at real histological material. But one of the questions we asked from our Autism Phenome Project is the cortex thicker? You might say, well, maybe there's been a dysregulation of the number of neurons that are generated, so the cortex is going to be thicker. And we do this by using a program called FreeSurfer that allows us to measure the surface of the brain and the distance down to the white matter. And what we find is that if you look at cortical thickness, the kids with autism, their brains are not any thicker. That's what's on left. What we do see is that the surface area is increased. So the undulations of the cortex, there's more surface area of the cortex, but it's basically the same thickness. And even when you look at the kids with the big brains, the thickness of their brains is not any thicker, or the thickness of their cortex is not any thicker. Again, what we see is that the surface area has increased dramatically. And in fact, it's really the kids with the big brains that drive most of the changes that we see in terms of total brain volume and the surface area. The boys with normal-sized brains, actually their surface area is not much different from the typically developing kids, okay? So it's that subset of 15% that are really sort of pushing a lot of the findings that people have seen thus far. Is the whole brain just thicker or bigger? Has it scaled up? Is that what's going on? Turns out that these colored pictures here, I'll just point to the one on the bottom right, every place that you see a colored area, that's an area that the surface has increased. And so the surface is increasing in places that we think are important for autism, places like the fusiform gyrus in the temporal lobe, the orbital frontal cortex and dorsolateral prefrontal cortex. So it's not everywhere, but you can see a lot of the brain is colored in. And we're just trying to struggle, with these maps now to try and draw some implications from them. Yeah. Some people have found that. We've actually analyzed it in an earlier study and found very small regions of the parietal cortex actually had not increased folding, but changes in the depths of the folds. So that's another area that people have actually looked into. Yeah. So I just wanted to say a little bit about fibers. And I think that this is an image of the fiber systems in the brain. I've just been talking about the volume of the brain, but you can now actually study pathways from one part of the brain to the other. You can do it in a lot of different ways. And we do it in our Kids with the Autism Phenome Project using diffusion-weighted tensor imaging. So we can actually map the pathways from one region of the brain to the other, and ultimately you can color code them. The brain doesn't actually look like this, but you can color code them digitally later on. And we're actually using tools that were developed by Brian Wandell's group at Stanford, and it's called the Automated Fiber Tracking Quantification. And if people in the audience are interested in doing this with their own subjects, I'd recommend getting in touch with Brian. He makes all these tools freely available, actually being funded by the Simons Foundation to develop something of this as well. So I just wanted to make a couple of points because there's been a long history of fiber bundle studies in autism. I think a lot of it has been confounded by this issue that I brought up right at the beginning. You're looking at adaptation and compensation rather than looking at the problems. But there's even more issues with fiber tracing. So we published a paper just at the beginning of this year. Ryan Johnson's a postdoc in the laboratory. He looked at these different fiber bundles in the brains of typically developing children, but in the age range of the Autism Phenome Project. Actually, they're the control subjects from the Autism Phenome Project. And here's the point here, that fiber bundles, these are just one of them. This is called the cingulum bundle, so that little cartoon is actually an image of a fiber bundle. And what the little squiggles down below show you are the different kinds of parameters that, and I didn't go into this, I can certainly go into this later, but axial diffusivity, radial diffusivity, and fractional anisotropy are just different measures of coherence of the fiber bundles. And what Ryan has shown is that, number one, boys and girls fiber bundles are developing differently. So if you look at the same time, that's what the blue squiggle versus the red squiggle, that they have different diffusion parameters at a particular time in life. And so what that means is that if you start adding girls and boys to a study, it's gonna complicate things. You really have to do a sex-based analysis if you're looking both at normal development or if you're looking at pathology. That was number one. I'm sorry? They're not glial cells. These are actually axons. These are bundles of axons. Yeah. So Ryan has actually looked at 18 fiber bundles in our group of kids with autism and found that a number of them show differences. So the main point I wanted to get across here is that this first chart, all of these fiber bundles here, and they're labeled the right uncinate fasciculus that connects the temporal lobe with the frontal lobe. It's shown in the middle picture, the yellow thing on the right. All of these bundles actually show differences, even in the kids who have the normal-sized brains, the kids with autism in the normal-sized brains, okay? So just how big your brain is not very interesting marker for autism. It's probably going to be the connectivity that we're seeing. And so we are seeing in the kids that have the normal-sized brains alterations of their connectional patterns, again, at a very young age. What's interesting here is that if you look, for example, let me just show you. So look at this pathway here. Most times what people will do will take an average of the parameters along the entire pathway. But what we found is by dividing the pathway up into 100 different segments, we find that there's only certain regions of the pathway here or here that actually show significant differences, okay? So a lot of people in the past have missed that pathways are altered because they've taken a grand average of the entire pathway, and it's much more subtle than this. And again, these are tools that Brian Wandel and his group have made available to us. But it just goes to show, just like genetics, as the next levels of sophisticated imaging tools come on board, you start seeing more and more detail. Yes, axons are running the whole length of this. So, you're so smart. So the question here, these diffusion parameters, people have thought early on that if you take a grand average, it's going to say something about the axons themselves, right? But it's not true. It may be true. But it's also the possibility is that the relationship of those axons to other axons are what are altered, because these are voxels that are large enough that are actually looking at the environment around the bundles, okay? So these measures of diffusion are really not looking at individual axons. They're looking at large bundles and their environment. And so many of these things that we see, we don't think are because the axons are bad. We think the axons are probably perfectly good, but somehow their relationship to other axons has changed, and that's changed their diffusion parameters, okay? That's where the field of diffusion imaging is going, looking at the environment. And one other point I want to make about this is that when you now look at the kids who have the big brains, there is actually more bundles that are altered. So here's one of the first characteristics where we can say there's something really distinctly different about the kids who have the big brain form of autism. They have alterations in the anterior thalamic radiation. For reasons that I can't understand, they have alterations in the left cortical spinal tract. Only the kids who have the big brain form of autism, okay? So we're still struggling with this. Actually, we're writing this paper right now, so our story is still emerging. Yeah?
So these are changes in myelin in small circulation?
So they can be changes in myelin, and often in degenerative disorders, people talk about changes in myelin. But it's not necessarily the case. It could be that there's local changes in the environment. And by the environment, I mean, the cell populations. It could be other axon pathways going through. You really are looking at a voxel that has a lot of things going on in it, okay? So it doesn't have to necessarily be the cellular aspects of or the axonal integrity or its wrappings. It could be what it's traveling through. Okay, if it's traveling through an edematous region, for example, that's going to change these diffusion parameters, okay? Okay, so I sort of said I won't going to skip this slide. We're still struggling to figure out the complexity of these different pathways. They all contribute to things that we think are important for autism, but again, we're still trying to understand, how these map on to some of the behavioral impairments that we're seeing with the kids. There's two more topics, and I think I can get through both of them. I want to tell you a little bit about a different part of the brain, non-cortical, subcortical region of the brain that's been highly implicated in autism. This is called the amygdala. It's the red almond-shaped structure there that's seen. When we first started the Mind Institute and I first got involved in it, there were 5 studies that had looked at the amygdala. 2 said it was too big, 2 said that it was too small, and 1 said that it wasn't any different. So I thought, well, we'd worked on the amygdala, maybe we should look at this and see what's going on. And with Cindy Schumann, who was a graduate student then, is now actually a faculty member at the Mind Institute, we did a study. This is not the Autism Phenome Project. This predates the Autism Phenome Project. So older kids, 7.5 to 18. And I won't go into the details because it was published quite a number of years ago. We do manual tracing of the amygdala. This is it shown in 3 sections. This is how you would do a study if you wanted to trace the amygdala in a brain of a child with autism. The first thing we found was that if you plotted the size of the amygdala versus the age of the child, there's this incredible slope. Actually, the amygdala plumps up by 40% between 8 and 18. And so when we first saw this, I thought, maybe we screwed up. Maybe, this because actually the total brain during this period of time is decreasing in size by, approximately 10%. But it turns out that Jay Geed at the NIMH had data that we replicated because they had done it earlier. So the amygdala is unusual that it goes through a pre-adolescent growth spurt and gets larger in volume by about 40%. And so then when we looked at the kids with autism in this study, turns out that their amygdala by 8 was already at an adult size. So the unusual thing was that they didn't go through this pre-adolescent growth spurt. It had happened already some time previously. So just as a cartoon, typical kids, typical boys in this case, their amygdala between about 8 and 18 goes through this growth spurt that increases the volume by 40%. This is unique. It's the only part of the brain that I know of that does this. Whereas kids with autism, by the time that we looked at it in this study, their amygdala was already at an adult size. So again, an indication that there was precocious growth of the amygdala. What we've been able to, and actually now there's been a whole bunch of studies, and what the studies show is basically if you look at young kids with autism, the amygdala is enlarged. If you look at adolescents, it goes either way. And if you look at adults with autism, actually the amygdala is smaller. Again, so the take-home message is that autism is a developmental disorder, and when you actually look will matter. We actually did a post-mortem study I won't talk about, but we found in people that were older who have autism that the actual number of neurons in the amygdala is decreased by about 13%. So, again, another process that we don't understand. It's too big early on. You look at an adult, it's too small, and there appears to be neuronal loss. Interesting. But I wanted to tell you what we found from the Autism Phenome Project. And again, we have younger kids now between 3, 4, and 5. And what we found was that even at time 1, at 3 years of age, the amygdala, which is shown in red, is 6% larger in the kids with autism than in controls. 1 year later, it's 9% larger. So what we detected was that this is the period where there's a rapid growth rate. And when we looked at that formally, what we found was that this is just the left and right amygdala, but whereas the typical amygdala was growing at 7.5% per year, the kids with autism were having a growth rate of 12.5%. And the amygdala, again, is growing at a rate much faster than the total brain, which is only growing at 4%. And at this time frame, both the kids with autism and the typically developing kids, their total brain or their cortex is growing at the same rate. I'm sorry? It is. We haven't studied it in girls because we haven't felt that we have enough girls. But Jay Geed actually found that there's a different trajectory in girls, which I think will be interesting to further study. Yep. So, yeah. Yes. Correct. Good. Thank you for so that was a good point. So this growth rate is you see it either in the raw data or you see it if you control for total cerebral volume. Okay, so this is independent of the total brain earlier on getting bigger. Okay, and it so again gets more subtle, and I won't go into too much of the details, but to say that when you analyze the whole population, what you see is that there's 3 phenotypes of amygdala growth. So let's just look at the right amygdala down the bottom in the middle. So what we found is that there's about 40% of kids with autism whose amygdala is growing at the same rate as typically developing kids, 40%. There's another 40%, that is the big red bar that has the 18.7 above, whose amygdala is growing over twice as fast during this period compared to typicals. And there's a smaller subset in green whose amygdala is actually growing too small. And interestingly, at this point, these are really young kids. We actually haven't seen anything behaviorally that maps on to these different amygdala patterns of growth. But my guess is that we'll see that when we start analyzing these kids when they come back and they're 8 to 10 years of age or even older. So my hypothesis is that the amygdala, first and foremost, contributes to the anxiety associated with autism. And our young kids at 3, you don't see a lot of, or at least you can't detect a lot of symptoms of anxiety. But as the kids get older, that's going to emerge, and we think that that's when we'll actually see these subphenotypes having some relevance to the behavioral phenomenology of the kids. That remains to be seen. Let me try and finish, because I'm going to be on time, actually. So, and I'm happy to answer questions afterwards. Okay, so so basically, that's all I'm going to talk about the Autism Phenome Project. We're looking at lots of different things. We're looking at the relationship to these kids' immune system. We actually found, for example, that the kids who, Jerry mentioned the autoantibodies, the kids whose moms have the autoantibodies, and so they were exposed to those during pregnancy, they actually their brains are the biggest of the big. And we published a paper on that. Christine Nordahl is the first author of that. So, whether it was the exposure of those antibodies that had something to do with the brains being that large, we don't know at this point in time, but interestingly, we have monkey studies where we've been trying to create a monkey model of the autoimmune condition. And the monkeys who we exposed to antibodies from the mothers, their brains turned out to be enlarged compared to age-matched controls. And the final corker was it was only the boy monkeys that had the big brains and not the girl monkeys. So that has to be replicated, but it's all very interesting. So if in the last 5 minutes, I want to talk about efforts to look even younger at children with autism, or children who may go on to have autism. And we did a study. The nice thing about MRI is that it's non-invasive, it's safe. You can scan very young children. Actually, there's now numerous studies where pregnant women are being scanned, with adequate safety precautions, you can do this, and it's entirely safe. We wanted to see whether there were any indications that in young children, 6 months of age, that they might have some signature that would be predictive of autism. And we did this study with Sally Ozenoff, who's the world's expert on infant sibling studies and early detection. And she had basically come to the conclusion, Ami's going to be coming next month, and he may actually dispute this conclusion. But her conclusion was that at 6 months, there was nothing behaviorally that you could see that would be predictive of a child going on to have autism. It was only at 12 months where things started emerging, and then even at 12 months, they weren't absolutely diagnostic. She and most clinicians that I've worked with don't feel comfortable making a final diagnosis until about 24 months or so. But she has these infant sibling studies, and for those of you who are not familiar with infant sibling studies, the advantage to them is that the general population prevalence of autism is 1%, 1 out of 100. So if you wanted to look at the early phase of brain development associated with autism, it's very inefficient to scan 100 kids and then see one that's going to go on to have autism. But if you are in a family that has a child with autism, the likelihood that your next child is going to have autism goes up to around 25%, so 1 in 4. Okay? So what's happened is there's many, many studies now that have capitalized on that and have recruited families who have one child with autism and started intensively studying. Now there's a study at the Mind Institute led by Irva Hurst-Picciotto where she's actually begun in the pregnancy and taken samples of the mother during the pregnancy, and then they study the child postnatally onto diagnosis, which I think is the optimal way to do autism research. If I had to do it all over again, that's what I would do. I would do the imaging studies even earlier because I think even at looking at 2-year-olds or 3-year-olds is already too late. We should have gone earlier. And I'll show you why in a second. So we, in the context of an infant sibling study that Sally Rogers was doing, we got some funding to do a 2-year study. We thought it was a pilot study. We were able to evaluate 55 infants, 33 who were high risk, meaning they have a child in the family with autism, 22 who are low risk, so infant siblings of typically developing kids. We did MRIs at 6 to 9 months, and then 6 months later and 6 months later. But Sally was evaluating these children starting at 3 months and every 3 months. So very high density of behavioral observations. And I should say that Mark Shen, who is the graduate student, really led this project. He did a terrific job. So we get neuroradiologists to read all of the MRIs to look for anything that's obviously wrong. And we didn't see anything that was obviously wrong with these young kids, but we kept getting these reports back of generous extraaxial fluid or increased extraaxial fluid or benign extraaxial fluid. And so Mark and I decided to look at whatever the heck this was, benign extraaxial fluid. And it's a little bit hard to see, but I'll try and point it out without a pointer. The top row is a typically developing kid at 9 months. And what you can see basically is that the brain completely fills the skull. Okay, it goes right up to the surface. But if you look at the 3 panels down below here, what you see is that there's space. I shouldn't touch the grass, right? Is Jim around? He's going to get mad. That there's space in between the brain and the dura. So you can see it at 9 months, but then it continues on in the next 2 scans. So this is where cerebral spinal fluid is there in between the brain and the skull. And this is what they were talking about, the radiologists. So and it's really obvious if you look at other kinds of scans. So this is called a T2 weighted image. And there's, again, a typically developing child up top. Now where the water is, where the cerebral spinal fluid is in white. And you can see one of these children who go on to have a diagnosis of autism. It's just a massive amount. So we saw this and we thought, maybe there's something to this. And then Mark decided that we had to quantify this because we can't go on these radiologist generalizations. We had to quantify it. So he came up with a strategy to do that. The red profile on the right is the extraaxial fluid, and he measured it in all the children in this study. And what we found was, so we have actually 4 groups. The kids who in red go on to ultimately get a diagnosis of autism at 24 and 36 months. We have a second group of children in green that have developmental delays but don't go on to have autism. And then we have 2 groups of kids who ultimately are typically developing, either from the high risk or from the low risk groups, okay? What we found, first and foremost, is that the level of extraaxial fluid in red was significantly higher at the first time point and stayed higher for the remainder of the time that we were analyzing the children. And there was a dose effect. So the more extraaxial fluid you had at 6 months, the worse the severity of your autism was at 24 months when you were diagnosed, okay? And that was true also for the longer that you maintained the extraaxial fluid, the worse your diagnosis was. So then the question was, are these kids having more cerebral spinal fluid in their skull because their brains are small? And it turns out, so we did the total cerebral volume, and it turns out that these kids who had the increased extraaxial fluid who went on to have a diagnosis of autism also have big brains, okay? So again, this is the red line here. This is total cerebral volume. At 6 months, it wasn't significant, but at 12 months, it was significantly larger than all the other groups and persisted at 18 to 24 months. Okay. So we published the paper, and we're looking at all those other things in these kids as well. We're looking at fiber pathways, we're looking at volumes and whatever. But this was what came out as sort of striking. And so we're not experts on hydrocephalus, we're not experts on radiology. So we started asking around, and it just turns out that the cerebral spinal fluid and its impact on the brain has all of a sudden taken off. People are really starting to pay attention to it. For a long time, people thought the cerebral spinal fluid was just a shock absorber, just to prevent your brain from being damaged when you get hit. But it's now known that it's carrying away toxic metabolites, trophic factors, and there's actually quite a few studies now showing that what is in the cerebral spinal fluid can actually modify the development of the brain. So there was actually a terrific paper just a couple of months ago showing in Science that the amount of extraaxial fluid, when you go to sleep at night and you have REM sleep, the amount of extraaxial fluid that percolates into your brain increases by 60%. And then sort of percolates back out when you wake up. And what is basically people think is that this is sort of the sewer system of the brain. So it's actually cleaning out toxic metabolites. So the question is, what happens if you have too much of this early on? Could it be that there's a slowing of the flow of cerebral spinal fluid? And could it be building up increased levels of these metabolites that might actually be toxic to brain growth? Again, we don't know. It's things that we're just looking at. But we want to try and replicate the phenomenon. So we've looked in our autism phenome project, now these are in much older kids, to see whether there's more of the cerebral spinal fluid. We're just finishing up this study. We have about 30 more subjects to look at. But even in our autism phenome project, where the kids are 3 years old, you can see that there's a huge scatter both in the kids with autism on red and the typically developing ones. But there's a significantly increased level of extraaxial fluid in the population of kids with autism on the left. And you can see some that are really extreme up there. The only association that we found at this point, and we're looking at a lot of them, is actually the kids, and there's only a small number, there's only 6 kids so far that have actually shown seizure disorders in our Autism Phenome Project. But they actually, the extraaxial fluid predicts whether the kids are going to have seizure disorders or not. So there's an association there. But we're looking at lots of other things. Where we're really going to hopefully hone in on subtypes is that Joe Piven, who's running the infant brain imaging study out of University of North Carolina, but has sites across all these different locations, has graciously allowed us to look at his data set. And now, rather than 55 kids, they have over 400 kids in an infant sibling study. And they did MRIs just like we've done them. And we're currently finishing off an analysis. And I'm not at liberty we haven't even finished it, so I can't tell you what the results are. But I can tell you that Mark has to finish his thesis in 2 months. So we'll know the results very soon. Again, this is the best part of science, right? When you find something that you weren't expecting, and there may be interesting implications, and we're just beginning to try and understand those. So the conclusions. First of all, I hope I've convinced you that there's not going to be one pattern of alteration in autism. There's going to be many, and we're just beginning to understand that. And it takes large samples, hundreds and hundreds of kids. It's sort of like genetics. And the geneticists are now up to tens of thousands, but I'm still happy with hundreds for MRI studies. But in order to be able to see the subtlety of different trajectories that lead to autism. I've become a firm believer that we need to study the autistic brain as early as possible with imaging because I think, again, a lot of the changes that will lead to autism are happening prenatally and immediately postnatally. And I also think, as I said at the very beginning, that when you start looking at kids or individuals further out, you're looking at adaptation. You're no longer looking at autism. Not to say it's not an interesting topic to look at adaptation later on, but it's a different topic than trying to find out what is the pathology of autism. And I think it's conceivable, and a lot more work has to be done because there's been so little imaging of very young kids. But given the fact that we're still looking for a biomarker of autism, and that MRI is relatively non-invasive. It's not cheap, but it's not everybody in this room had one, so most medical care will actually take care of it. That MRI could actually be a valuable adjunct, a clinical adjunct, to increasing our confidence of increased risk. And of course, the name of the game in autism is to be able to develop some kind of information that will allow a family to know that they should go into early intervention and take on the cost and the difficulties with doing that, because we know that early intervention will lead to the best possible outcome. So again, thanks very much. This is my home, and I'm happy to answer questions. Thank you.